The floor is yours, Ondra. Thank you very much.
Thank you, Honza. I hope everyone can hear me. If not, just give me a shout. And as Honza said, I will talk about our recent paper about Salmonella. But yes, I will describe the results published in the paper, but I hope that it will be more like tool related to you all. So if in case you see some inspiration for the tools, we are happy to share all our knowledge and tools we generated, and um, in this line, everything. I guess that's more important for now than the real results. So what we do, or what's our most interest in the lab, is studying the heterogeneity of Salmonella and the cooperation among individual bacteria inside host cells. So most of this was done by Milada, and to bacteria now. If we start with a bacterium, I imagine something like this. I don't know, what do you imagine? But what's important is that this bacterium can replicate and divide over time. So you have 2 bacteria after one replication, you would have 4, and so on after several replications. So I just stop here by putting 3 bacteria on the slide. And usually people consider these bacteria originating from single CFU as, let's say, clonal identical organisms. But what's real is that each of those bacteria behave differently and express slightly different set of proteins or different set of genes during the cultivation. So back to Salmonella. As you know, Salmonella is a pathogenic bacterium. It's gram-negative rod-shaped bacteria that can or do not have to produce flagella. So at certain conditions is motile, and it causes either gastroenteritis, manifested as diarrhea, or typhoid fever, which is basically systemic dissemination of the bacteria or bacteremia, septicemia in the host. So then it's facultatively intracellular, so not necessarily living inside the host cells, but most of the clinical manifestations are associated with bacteria living inside host cells, but not necessarily as depicted in here, freely in the cytosol, but it's mostly encapsulated in sort of modified phagosome or endosome that we call Salmonella containing vacuole, or SCV. And it's quite small place, and it's surrounding Salmonella quite tightly. And to survive in this vacuole, Salmonella has to express type 3 secretion system encoded on Salmonella pathogenicity island 2 that we abbreviated SPY2. So if you hear me at some point talking about SPY2, then I mean this type 3 secretion system encoded on Salmonella pathogenicity island 2. And what this type 3 secretion system is that's basically a small nanomachine syringe that punches a hole in the membrane of the host, in this case in the membrane of the Salmonella containing vacuole, and it pumps small proteins from the cytosol of the bacterium straight into the cytosol of the host cell. So no secretion into the periplasm, no secretion into the lumen of the vacuole, straight into the cytoplasm of the host cell. And these small proteins are then called effectors and can have several functions. And here I put 2 different ones. This blue one is called CIF-A, and it's important for Salmonella survival in the vacuole and for synchronization of division of the vacuole with division of the bacterium. And this green one is called STED, and it has nothing to do with Salmonella survival in the host cells. It manipulates T cells response. And the function is actually not that important for this paper. So then, as I said, you probably have more bacteria, so 2 or 3, and they all look identical on the first look. But the question is if they really are identical or if they are not, even when they originated from the one individual bacterium at the beginning of the infection. So to answer this question, we constructed Salmonella that carries M. Sherry or express M. Sherry under the control of constitutive promoter. And in all our experiments, this is located on chromosome and helps us to identify the bacteria in whatever sample we have. Then we also have green fluorescent protein that expressed under the control of a promoter originating in the Salmonella pathogenicity island 2. So this helps us to monitor the activity of the Salmonella pathogenicity island genes. And it can be located either on chromosome or on plasmid. In all the experiments we did, this basically behaves the same. And then we may have a third fluorescent protein that would monitor expression of the individual effectors because they are not located in the pathogenicity island itself, and their expression may be different from the expression of the pathogenicity island. And this may be blue as on this schematics, or it can be green as well, depending on what combination of the pathogenicity island promoter or effector promoters we are using in the experiments. So in theory, we would be able to distinguish different populations of bacteria. The pink one, expressing only M. Sherry, would not be expressing either the SPY2 and the type 3 secretion system, nor the effectors. Then we can have bacteria that would be expressing GFP, and those would have active expression of the SPY2 genes, and potentially bacteria that are combination of blue and green, plus M. Sherry, plus pink as well. And these would have not only active expression of the secretion system, but also of the chosen effector. And at the first experiment, I will focus just on the secretion system itself and comparison between these two states of bacteria. So first we infected epithelial cells with Salmonella that's always pink and green when expressing the secretion system. And then after some time, we monitored the bacteria inside host cell. And you can see basically exactly what I was describing on the previous slide. So inside one epithelial cell, you see several bacteria. All of them would be pink, as seen in here. And in the overlay image, you can see that some of them are more green, some of them are less green, that would turn white in the combination, and some of them are only pink. So these two bacteria do not express any GFP, means that they do not have active SPY2 secretion system. And now I need to make one step back and remind you that the expression of the SPY2 secretion system is crucial for Salmonella survival inside the host cells. And I will come back to this a little bit later. So what I wanted to say is that here, if we quantify these bacteria using flow cytometry, we would find a population of bacteria inside the same host cell that does not express the crucial type 3 secretion system, and we call these bacteria SPY2 off. And then you also have a population of bacteria that do express the type 3 secretion system, and we call these SPY2 on bacteria. That was about the secretion system itself. So now if we go back to the effectors, in this experiment, we took again the always pink bacteria and expressed BFP under the control of one effector promoter and GFP under the control of the other effector promoter. And after some time, we would expect to have a mixture of bacteria of all three colors. And here I need to promote the confocal microscope we got from this OPIAC project, Stellaris 8. If you want to give it a try or use this confocal, please contact someone, me or Jana or whoever knows how to work on the confocal. It's really a nice microscope and can you give really nice data and nice pictures. So here again we see one single epithelial cell infected with many different bacteria, and in this zoomed area you can again see a mixture of different population or differently behaving bacteria. Again, all of them are pink. And for example, this one is almost only pink. There is no expression or very little expression of one of the effectors and very little expression of the other effector. But in here, let's say, you have very little expression of the second green effector, but very high expression of the blue effector, while in this bacterium have very low expression of the blue of the blue effector and very high expression of the green effector. So exactly as depicted on the schematics, you can really get a different behavior of all the bacteria inside the host cells, even though they are clonal. And this is not only the case in a test tube, but the same we can see after infecting of black 6 mice. So when we feeded them with Salmonella and then isolated bacteria from mesenteric lymph nodes, we again saw that there is a population of bacteria not expressing either of the two effector proteins, and some bacteria that express very high amount of both of the proteins. So to the regulation of these effectors or expression of these effectors and of the SPY2 itself. And now again, I would like you to think about this as a proof of concept that these can be used to study expression of genes in bacteria in the real world. So the SPY2 genes are activated by SSRB response regulator after it's binding on the promoter of the SPY2 genes. But then SSRB has to be phosphorylated to be active by a sensor kinase, SSRA. So this is the canonical two-component signaling system in bacteria. And for us, the question was if these proteins are those that do regulate the heterogenic behavior in the clonal bacterial population. So we took again always pink Salmonella that would turn green when expressing the SPY2 proteins. We infected macrophages now, and we again observed the green and pink ratio in the bacteria after some time. And as you would expect already, using the wild-type bacteria, we had bacteria that did produce the SPY2 type 3 secretion system and bacteria that did not in the same host cells. When deleting the response regulator, as you would expect, there is no expression of the SPY2 genes. And then when we reintroduce the gene back under the control of a constitutive promoter, so all bacteria do express the system, we again saw a renewal of the positive peak, but still some bacteria remaining negative. So just the crosstalk in between the kinase and the regulator in this system did change the ratio in between negative and positive bacteria. But then we also deleted the kinase. So in this part of the slide, we have both proteins missing. And again, as you would expect, the deletion leads to complete stop of expression of the SPY2 genes. But then even in the absence of the kinase, when the regulator is expressed constitutively in all bacteria, you get very weak but homogeneous, and that's important, homogeneous activation of expression of the SPY2 genes. So it's really the crosstalk and phosphorylation of the response regulator by the kinase, by the sensor, not just the presence of the protein that regulates the bimodal behavior. Quite funnily enough, this paper was published in December, and I think the last week was shown on bioRxiv, a paper that shows exactly the same. Plus, they also calculate some affinity between the two proteins in the different stages. So I am now confident that it's really true. So yeah, that I already said. I don't really know the group, but they are from the US. We scooped them, yeah. Yeah, yeah. So back to the presentation, sorry. Now we know that the bacteria do behave differently, either even though they are clonal in the host cells. And the question would be how this is relevant for the physiology of bacteria inside the host cells. So we now took wild-type bacteria that are only pink and mutant bacteria that don't have the SPY2 genes and they are only green. So in this experiment, we only have green and red bacteria. And I should say one limitation of this experiment is that these bacteria can be both SPY2 on and SPY2 off. So we don't have a real distinguishing between SPY2 off and on, but we know that these are always off. And then we infected a single macrophage with both bacteria and observed the survival of these green guys, because as I said already twice, the bacteria need to have the SPY2 to survive in the host cells. So in theory, these bacteria should not be able to survive and should not proliferate in the host cells. So when you observe a macrophage and all these come from the same well, so it's not like batch effect, let's say. So the wild type, the pink bacteria, even though there was just one bacterium at the origin, at the beginning of the experiment in the macrophage, these bacteria do proliferate. Then when you have the mutant, the bacterium is still there, it's still living in this case, but it does not proliferate. And when you have both of them, you can see that the wild type, yes, it proliferated quite a lot, but also the mutant did proliferate. So that means that the SPY2 on bacteria coming from the wild type, whatever fraction of these are SPY2 on, managed to protect these always SPY2 off green bacteria and enable them to proliferate. And what's also interesting, and we don't have any explanation for that yet, is that actually these green bacteria, maybe these ones, do help in certain amount to these guys. And we don't know, we don't have any explanation for it yet, but there is a statistically significant more bacteria of the red bacteria when the green ones are there as well. So there is a real cooperation in between the SPY2 off and SPY2 on population. So one way, if you imagine that those bacteria, the SPY2 off or mutant that I showed you on the slide before, grew quite a lot, then how would we monitor how quickly they grow? So we took now a system where we have the bacteria blue when they are the SPY2 positive, and green in case they found some arabinose around them and they respond to the arabinose. So we gave them arabinose in the medium and we observed all the bacteria turning green, as you would expect. And then we used these bacteria for infecting macrophages. So at the beginning of the infection, all bacteria inside the same macrophage were very highly green. And then as the infection continues, we expected to see how the green color dilutes in the dividing bacteria in analogy to T-cell proliferation assays in immunology. So you can see that after 6 hours post-infection, we have there green bacterial population that's not expressing the type 3 secretion system. And it's known from Salmonella that there is always a fraction of bacteria that, even though living and metabolically active, do not proliferate. And these bacteria are actually antibiotic-resistant persisters. Then we see here a population of bacteria that has no fluorescence at all. And these are originating from this population and lost all the GFP signal already because they divided very much. And then you have here a population of bacteria that expressed the secretion system and start losing the GFP fluorescence, but not that much as these guys, meaning that they proliferate slower than the spy2-off bacteria in here. And when you keep the experiment running for a little bit longer, you see that these spy2-on bacteria catch up with diluting the GFP signal over time. So these spy2-on bacteria proliferate slower, and these spy2-off bacteria proliferate faster. And the question then is if, again, this speed of proliferation has some physiologically relevant benefit for the spy2-off bacteria and for the conservation of the bimodal behavior in evolution, because you could expect that over time of evolution, the bacteria would fix either one or the other phenotype if it's not mutually beneficial for both of them. And it's also known that when the host cells contain a lot of bacteria, they just burst and the bacteria can escape from the dead host cells. So we again took the always pink bacteria, turning green when expressing the spy2 genes, and we monitor the expression of the GFP reporter in the macrophages that contain living bacteria, in living macrophages containing bacteria, and in dying macrophages that are just releasing bacteria in the medium around the cells. So we can just harvest the supernatant in this culture, and we would get bacteria that escaped from the dying macrophages. And again, the same scenario as I already showed you before. In the living macrophages, you have a population of spy2-off bacteria and a population of spy2-on bacteria. But then when you take the bacteria from the supernatant, you see almost only bacteria that are or were spy2-off, and very little bacteria that were spy2-on at the time when leaving the macrophage. So what we think is that the bacteria change the expression of the virulence genes in the macrophages, in forming the different subpopulations, and then those that were spy2-off proliferated faster and reached the critical mass of the bacteria inside the infected cell faster and killed the macrophage faster, and then enabled the bacteria to spread around to new host cells. But for this, they still need few spy2-on bacteria that allow them to survive in the host cell. So in case you find this interesting, and if in case you think you can use something of this in your essays or in your projects, we would be happy to share our reporter systems or the knowledge we accumulated regarding these reporter systems, usable also for signaling in the bacteria and in the host cells, and our knowledge on flow cytometry of bacteria and confocal microscopy of the bacteria, and we will be happy to teach you about the Stellaris 8 we have now in here. And that's it. Thank you for your attention.